Welcome, Biology 220. Today we're going to be covering the microbiology notes. Microbiology is the study of microorganisms, or what we can call microbes. So, what defines a microbe is an organism that is so small that it must be viewed with a microscope. And we'll look through examples of all of our microbes today. First example of a microbe would be a virus. So virology would be the study of viruses. So example we'd all be familiar with right now would be COVID-19. Also probably remember when Ebola was in the news a lot. Hepatitis C, rabies, which we'll talk about some here today. So for viruses, they are still going to be considered microbes because they are microscopic infectious agents. But they are unique because they would be acellular. They do not have cells, and they must have a host to reproduce. So, they kind of blur the line between living and non-living things, which would be different from the cellular microbes like bacteria and fungi. And these are going to be, for viruses, that's going to be the smallest microbes that we'll look at. So you have bacteria as a microbe, and bacteriology would be the study of bacteria. And here are some different shapes that we'll actually look at in the lab, right? Coccus, bacillus, spirillum, and we'll get to look at all these under the microscopes in this course. Protozoology, that will be the study of a protozoa. So for example, amoebas, euglena, paramecium. We'll get to look at those. I believe it's the lab 4, living materials lab. Really cool to look at under the microscope. We get to observe them while they're alive. So you get to see them swimming around, or the amoeba crawling around with its pseudopodia. We have phycology, and that's going to be the study of algae. Mycology is going to be the study of fungi. Now, we think of bacteria as all being problematic, but only 3% of all microbes are actually pathogenic in all these categories on average. So most of them are not going to cause any problems for you. 97% would be either non-harmful or beneficial to us. So here are just some examples. I'm not going to go through all these, but different examples caused by these different types of microbes. All right, for viruses, you've got rabies, polio, different bacterial diseases, syphilis, gonorrhea, food poisoning, strep throat. Protozoans could be malaria, fungi, yeast infections, and then helminths could be pinworms, tapeworms, things of that sort. So again, 97% of these microbes are going to be non-pathogenic, and they provide lots of benefits that we'll actually look through right now. If you remember from Biology 103, we talked a lot about cellular respiration. Cellular respiration is the process where we're able to create ATP from ultimately glucose. Remember, we get through glycolysis and then all the cycles until we eventually get to the electron transport chain, and then we produce a massive amount of ATP. But that only happens in the presence of oxygen. If oxygen is not available, then it goes into something called fermentation. So bacteria, fungi, they like to ferment sugars during this process to create ATP, and they'll produce, it is used in the process of beer making, wine making, and fermentation for even like sourdough breads, cheese, and yogurts, which is pretty interesting. So the benefits of microbes, one of those would be yogurt production. So it's got to have a sugar source even to do fermentation. So the milk has the lactose. Lactic acid bacteria are added to the milk in the yogurt production process. The bacteria feed on lactose, and then they go through fermentation, and that's going to produce lactic acid, which is going to create an acidic situation. And the milk protein casein will thicken and coagulate to produce yogurt. And that lactic acid gives it that nice sour flavor people like. It's also used in making of cheese. Microbes are used in the making of bread, like yeast, to make the bread rise. The yeast is going through fermentation. Then in wine making, you just need a sugar, so the sugar comes from the grapes, and then the yeast is going to provide the process of fermentation, and as a result of that, you're going to get alcohol and carbon dioxide. So it naturally carbonates it, actually. Same thing with beer, but with beer, rather than just using grapes, it's going to get the sugar for the process of fermentation from barley, and then hops are added for flavor, and it's actually a preservative as well. So it'll get the sugar from the barley, flavor from the hops, and then it goes through the process with yeast again for fermentation to get the alcohol, which would be in the form of ethanol and CO2. Some other benefits of microbes would be in the development of medicines. We think about penicillin. Penicillin is actually produced from a fungus called the penicillium. It was actually discovered in 1928 by accident by Alexander Fleming, who basically he created a culture, a Staphylococcus bacteria culture, and when he came back to it, it had been contaminated by accident with a mold. And when he looked at the petri dish, all around the penicillium fungus, there was no bacteria able to grow. So there was that zone of inhibited growth. And so he hypothesized that it was producing an antibiotic, something that is anti-bacteria. So in the development of vaccines, microbes are super important. If you want to develop a vaccine against a certain virus, well, you can take that virus and attenuate it, make it weak, and then you can give that to somebody, and it's a weakened form of the virus, so it can't harm them, but their immune system can still interact with it to develop antibodies against it. So they're able to build up immunity and memory against that virus in the future, and you can do the same thing for bacteria. Bacteria are also really important in food chains as well as in oxygen production. You know, there's bacteria that produces, if I remember correctly, most of the oxygen for the planet is from bacteria in the ocean. And they're also a good food source at the bottom of certain food chains. We also have different microbes that play a huge role in decomposing materials and recycling nutrients. And then in aquatic environments especially, they are usually going to be at the foundation of them. We'll see them for food chains. They produce oxygen, they can reduce carbon dioxide, and they can serve as decomposers. They're also able to, certain bacteria are able to fix nitrogen through a process called nitrogen fixation, which is super important for life on Earth because N2 molecules are really hard to get out of the air. So certain bacteria that live in this symbiotic relationship with plants and these little root nodules, the plant grows these little homes for the bacteria, and they live in these nodules around the plant roots, and then they are able to take diatomical nitrogen into and break that triple bond and convert it into absorbable useful forms. So plants can absorb that nitrogen and use it to make DNA. So without that process happening, we wouldn't be able to get the nitrogen we need to be able to build DNA and make new cells, to even be able to go through mitosis. Before that happens, you got to go through S phase, and you need a lot of nitrogen to make all that DNA. Some other benefits would be bioremediation. So if you remember, you may not remember, but not all that long ago, we had the BP oil spill. And when that happened, they were trying to figure out all these ways. Of course, initially it took a couple months, but to plug the leak. But they also wanted to figure out how to get rid of all this pollution, all the polluting oil. And they've actually designed bacteria, and even natural bacteria in the environment can be stimulated to break down the oil droplets into smaller pieces. So they call that bioremediation. And they use their enzymes, the bacteria produce their own enzymes to be able to break down oil. And the products of that oil, when it's broken down by the bacteria, were actually not harmful to the coral reefs like the large initial oil that was released. How it was harming the coral reefs, it broke it down, and it was to less harmful or non-harmful substances. So the BP oil spill was called the Deepwater Horizon spill, and that was April 20th, 2010. On the left, you see a brown pelican covered in oil from that spill, and that was on the right, someone reaching in to just demonstrate how thick that oil was when it came through. They actually, to stop it, they tried to like cap it with these temporary caps, and it wasn't working, and then they eventually got a permanent large cap over the wellhead, which was pretty crazy. So it took them 87 days to stop the leak. So other benefits, it can be used for water purification, where you can get the microbes to eat up pollutants, just like we looked at with oil and other toxic substances that might be in the oil, so they can be safe for drinking. Microbes are used through genetic engineering to actually change the DNA in organisms, and we can actually change the DNA in microbes themselves, in bacteria, for example, to give them the genes to produce proteins that are useful to humans. So, for example, with bacteria, this is still how the majority of insulin is made for people with diabetes around the world, is through genetic engineering of bacteria. They're able to use these restriction enzymes to cut into what's called a plasmid, and we'll actually talk about this more at a later time. But they use restriction enzymes to cut a part of a plasmid, which is an additional piece of DNA that bacteria can have. And then they'll actually put in a human gene for insulin protein in the plasmid the bacteria had. And then they put the plasmid back into bacteria. And the way you can make bacteria release and absorb things is heating them up and cooling them down. Well, once they absorb the plasmid, then you can give them the nutrients they need to reproduce. And then every new cell that's produced will also be reproducing their plasmids. So you get this huge population, and fermentation helps aid in that process of reproduction. Huge population of bacteria that can produce insulin. And then you just stimulate them to produce insulin. And usually you can put something upstream of the gene, like a food source that triggers the turning on of that gene. So they'll produce insulin when they're given a certain thing to metabolize. And then they'll just produce a bunch of insulin. It'll build up on top of the bacteria, literally in the petri dish or whatever container they're growing in. And then you can just collect that insulin, purify it, and then distribute it. And they used to use insulin from pigs and cows, but the problem with that is people would have severe allergic reactions a lot of the time. But if we're using a human insulin gene, then it's perfectly complementary in our bodies, and it works very well. And here's just another image of what that recombinant DNA looks like. So it's literally bacteria through this process that is keeping millions of diabetics around the world alive right now. And again, remember we said that only about 3% of bacteria are pathogenic, 97%, or 3% of microbes are pathogenic, 97% are going to be non-harmful or beneficial. And in our own bodies, where the non-harmful bacteria or beneficial bacteria plays a major role, is taking up space. So, for example, if bacteria are trying to infect your body, pathogenic bacteria, like in this image here, well, if all of the places to live are already taken up by good bacteria, then the bad bacteria can't find a place to take up residence. So they're basically excluded because all the good places to, get settled in are all taken up. So what can happen is, somebody takes antibiotics for a while, and then they're exposed to something. Well, all the good bacteria is dead too. So then you're more susceptible to a pathogenic bacteria afterwards. So what's really good is to eat probiotics while you're on antibiotics, and it reduces diarrhea-associated antibiotic usage. It reduces that diarrhea you might have with it, and it can help prevent infection from another bacteria or pathogenic bacteria. So now we're going to talk about milestones in microbiology. Robert Hooke will be our first scientist we'll look at. From 1635 to 1703 is when he lived. He was a physicist and a chemist, and he's considered the father of microscopy. And he's the one who's famous for giving us the term cell and cells that we use when we're talking about organisms and histology. So if you look at the image on the left, all those empty squares is what he drew. He drew that. And he called them cells, named after what his observation or imagination of a monk's study cell would look like in a monastery. So if you think about the little square room that a monk would study scripture in a monastery, he said that these look like that. So he named them cells. It's pretty interesting. And the microscopes were actually invented about 50 years before his studies, but he was really the one who popularized them and made significant discoveries using them, but he did not invent the microscope. Anton von Leeuwenhoek, from 1632 to 1723, used microscopes, and he was a biologist, and he's considered the father of microbiology. So he actually was reading Hooke's works, and they inspired him, and he actually ended up making 400 different types of microscopes, just improving them and changing them in different ways. And he's the one who named his observations animalcules. And he named them that when he looked at pond water, because these little organisms, they looked like molecules, but they moved like animals. So he called them animalcules. He did not believe in spontaneous generation, which is something we're about to talk about, which would be the idea that, living things just come from non-living things, that life just spontaneously generates. And his experiments to, aid in the disproving that was that he looked at these tiny animals and insects, and he found that the offspring were coming from parents, and they were coming from eggs. We get to Francisco Redi, 1626 to 1697. He was a physician, a biologist, and a naturalist, and he ultimately disproves spontaneous generation for flies. And this is his little, experiment. If you take, and this is why people believed in spontaneous generation. If you just put, a piece of meat, even in an area that was pretty controlled, eventually it would have flies on it. And they thought, well, maybe the flies are coming from the meat. So what he did was he took one glass jar and he put a piece of meat in it. It was open to the environment. And eventually flies came in, and eventually maggots were produced, and you get more flies as eggs are laid and what have you. Well, then he got one and he put gauze over the top. And when the gauze was over the top, nothing could get in. So it couldn't produce anything they couldn't lay their eggs in the meat or eat the meat. So it prevented the formation of flies and maggots on the meat. And then they did it with parchment, and you get the same thing. So for this, he said, well, life comes from non-life. If there's not anything living, then you're not going to get anything living from it. John Needham, 1713 to 1781. He was an English biologist and Catholic priest. You'll notice a lot of these early scientists were actually Catholic priests or associated with the Catholic Church, or were even maybe funded by the Catholic Church, because of course that was the dominant religious affiliation in Europe at the time. And a lot of times, if you couldn't get money from them, you wouldn't have the time or the money to do the studies to begin with. But he believed that microbes spontaneously generated. Because if he took broth, eventually microbes would grow in that broth. Even if you boiled it, if you left it out long enough, bacteria would grow in it again. And you'll notice with his flask here, it is open to the air. So then we get to Spallanzani, and he was also against spontaneous generation. And in his experiment, he noticed that the microbes had to come from the air. So if you close the flask, right, after it's boiled, while it's boiling, well, everything dies, and then nothing can get in from the air. Then we get to Antoine Laurent Lavoisier. That's how you say that. From 1743 to 1794, he was a French chemist, and he is considered the father of modern chemistry. He was able to separate water into hydrogen and oxygen, and he discovered that most living organisms need oxygen to survive. Then we get to Louis Pasteur, who lived from 1822 to 1895. This is probably one of my most favorite scientists in all of history. He has some really awesome experiments and some cool stories associated with him. But he disproved spontaneous generation of microbes in a broth using the curved neck of a flask, which is pretty cool. So he noticed bacteria spoil wine. He developed the process of pasteurization, which is rather than boiling something super hot, you get it to a moderately warm temperature to kill most microbes, which extends the life of milk significantly, but it doesn't destroy everything. He said that microbes cause disease and help develop germ theory, and he also developed the rabies vaccine. So look at a few things from him, but first I want to start with a story about Louis Pasteur. This young man in this image with Louis Pasteur is Joseph Meister. He was a 9-year-old boy at the time when he meets Louis Pasteur and meets him under less than ideal circumstances. He was bitten 14 times by a rabid dog. And everybody knew there was no solution, no cure, nothing that could be done for someone who was bit by an animal with rabies. So the family, everybody thought, well, Joseph, our son, our 9-year-old boy, he's going to die. Everyone who gets bit by a rabid animal dies. But Joseph Meister's parents, they were looking through some newspaper reports, and they noticed this story about Louis Pasteur and that he had been working on a rabies vaccine. And he had developed it effectively for dogs, but it had not been used on humans yet. So they actually reached out to him and tried to ask him to help with their son who had been bitten by this dog. Well, rabies as a virus, it takes about 2 weeks to multiple months, depending on how long it progresses, but it could be as little as 2 weeks. But it moves from the peripheral nervous system to the central nervous system. So if somebody can get the vaccines before it reaches their central nervous system, then they can actually develop immunity before it gets there, and it can save their life. So they gave the vaccines to Joseph Meister, and it eventually did give him immunity before the virus got to his central nervous system and saved his life. And the way Louis Pasteur developed that vaccine was he took the spinal cords of animals that were infected with rabies, and when he extracted them from the animal, if he left them, he had a drying compound, and he would leave them hanging, covered in this drying compounds, then the longer they dried out, the less virulent they were. So he could introduce an animal to the less and less virulent strains and up to the more virulent until the animal became immune. And that's what they did with Joseph Meister. And he lived. And he actually became like a huge fan of Louis Pasteur, obviously, after he saved his life. And he actually started to work for him at his institute. And sadly, he actually committed suicide in 1940 because he thought the German soldiers were going to destroy the institute that he worked for and the tomb. It was really sad because they actually didn't. And he killed himself really for nothing. But he was eternally grateful to Louis Pasteur for saving his life. So Pasteur developed something called the curved neck flask experiment to disprove spontaneous generation. So what they took was a non-sterile liquid, and they poured it into a flask. Then in the flask, they had this curved neck on it. And when they heated it up, it forced the air out the open end of the flask. So the liquid is completely sterilized through the boiling of the liquid. Well, what would happen, because the neck was curved, any dust and microorganisms that may be in the air, they would get stuck in the curve of the flask. So when the liquid cooled, these dust and microorganisms would get in and they would get stuck. So what happens is the liquid always remains sterile. So if you took the flask and tipped it over so that the liquid would touch the microorganisms, and then if you tilt it back, well, then the solution would become infected with microorganisms. So he proved that they were stuck in the curved neck of the flask. So it disproves a lot of misconceptions leading up to his experiment all at once. It was pretty cool. And then pasteurization he also developed, and that rather than boiling, you use a mild heat, and rather than killing all the microbes, it just kills some of them, and it prolongs the shelf life of the milk. So the contraptions often involve these long tubes of moderate heating to get rid of a lot of the microorganisms. It also keeps the flavor largely intact. It doesn't make it taste terrible. As if you boiled it, it would really make it taste not very good. Then you get to Robert Koch, 1843 to 1968. He was a German physician and microbiologist. He actually was able to correlate the cause of tuberculosis, cholera, and anthrax. And what you'll have to learn from him is his 4 principles, what's called Koch's postulates. And this is how you can always identify the specific microbe that is causing a specific disease. How you can be sure that that is the microorganism that is causing that specific disease. So we'll go through Koch's postulates. His first postulate says that the microorganism must be found in abundance in all organisms that have that disease. Right? If we're going to say this microorganism causes a disease, well, it has to be in the animals that have the disease. If it's not, then those animals can't have that microorganism. That microorganism can't be causing that disease. So that's the first one. The animal has to have the microorganism. Number 2, the microorganism must be isolated from a diseased organism and grown in a pure culture. So you want to know that it's in there. You want to be able to get it out and have it separated from the organism. The third part of Koch's postulates would be to take the cultured organism and introduce it into a healthy organism. If you take it, right, you took it out of an infected animal and that animal had the disease. You got it in pure culture. Now you're going to give it to another healthy animal. If it causes the disease again, well, then you get to Koch's 4th postulate. The microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. So then you have to be able to get it out and see, is what we're looking at now that killed this organism or caused the disease again, is it the same as the microorganism that I isolated the first time? If it is, you know that is the microorganism that caused that disease. Really, really well thought out, very practical. Then we get to Edward Jenner, 1749 to 1823. He was a physician, and he performed the first vaccine. And what he did was he took a related vaccine to vaccinate against another more deadly virus. So cowpox was around, and he used that to vaccinate humans against smallpox. So he would give them cowpox, and then they would get immune to smallpox. And this was actually decades before Louis Pasteur. But what makes Pasteur considered the father of immunology as he is he actually developed the lab procedure to consistently develop the vaccines. Whereas Jenner, he would just take, he would just take the virus and give it to somebody else. It wasn't the same sort of well thought out, repeatable lab setting experiment. And this is smallpox here in the images. So you can see all these little bumps that it creates. It's actually a horrible disease. So the symptoms for smallpox, it would start, you'd have a fever, then it would get to a high fever. You would get a headache, body aches. You'd be super tired. Then you would get a rash, and it would start in the mouth, and then it would spread to the face, the arms, the legs. Then you would get the fluid-filled blisters, they call them vesicles, and then pus-filled sores, pustules, and then finally it will crust over into scabs, and they'll leave scars on their skin. Extremely contagious. It goes through different stages, and the rash appears about 2 to 4 days after you get the flu-like symptoms that you would have at the beginning, and it'll be weeks before the scabs fall off. Depending on what strain that you have, it could be extremely deadly or not very bad at all. So one of the strains has about a 30% fatality rate. It would be over 50% in unvaccinated people. And then milder strains might be less than 1%. And then there was a crazy strain that actually had 100% fatality. But anyway, how he developed the vaccine was the milkmaids who were exposed to cows would get cowpox, get on their hands, but then those individuals would not get smallpox. Or if they did, they wouldn't have bad symptoms at all. So what you could take is the pus from cowpox, a milkmaid who had cowpox, give it to somebody else, they get cowpox, but then they don't die from smallpox. That's essentially what happened. They would transfer the cowpox pus, and then they would give it to somebody else. So smallpox did kill millions of people. And the people who survived had horrific scarring and these little pits all over their skin. Certain individuals genetically were less resilient to smallpox. So Native Americans, for example, they were extremely susceptible to smallpox. So if they got it, they were very likely to die. So now what we've done is there was an eradication campaign, and the entire world essentially were vaccinated, and it was actually considered, it is considered eradicated. It does not exist in nature anymore. That happened in 1980. Now, because the United States and Russia, we had the Cold War, and because neither of us trust each other, we have both kept smallpox virus in our facilities, in our labs. So we have it and Russia has it. So if Russia tried to create, some type of viral epidemic in the United States using smallpox, because we still have it, we could develop a vaccine if we had to. But people do not get smallpox vaccines anymore because unless we're in a germ war, we're not going to need it. So anyway, that's someone giving the smallpox vaccine. You can see what it does to people's bodies. It can be a very brutal disease. So this is an image from 1947 on the right. You see this huge line of people in New York City. And they actually created a campaign to give everyone a free smallpox vaccine so that they wouldn't die from the disease and to prevent the spread of the disease. So, at that time, everybody wanted to get that vaccine because it was a life or death situation. Could be. Then you get to Joseph Lister, also a really interesting scientist. It's 1827 to 1912. And he was a surgeon, and he was the first person to use antiseptics in his surgical procedures. And he actually was reading a lot about Pasteur, and he was familiar with his works that showed, fermentation and decay were caused by microorganisms. So he thought, well, maybe microbes are also causing these infections like sepsis, gangrene, that are killing his patients. So he thought, if we can get things to kill these microbes, maybe these people will live through their operations. So one of the things he did is he used carbolic acid, and he would use carbolic acid covered gauze over the incision points and things of that sort. And he noticed a dramatic decrease in fatality. So the mortality rate fell in from his surgeries from 47% to 15% in his ward, which is very good improvement there. Then we get to John Snow, 1813 to 1858. He was an English physician, and he's considered the father of epidemiology. And he actually traced a cholera outbreak through asking people are going to people who were infected and figuring out where they lived, and he created a map of infected individuals, and they were all around, just draw a circle around this well. It was a well pump, actually. So he said, okay, this cholera is actually being spread from people at the well pump. So then they started to encourage everyone to boil their water, and then it was able to cut back on people getting cholera. And cholera, it's a disease where you just have really intense diarrhea. You lose all your electrolytes in water. So then people are dying of dehydration. That brings us to classification. So for our types of microbes, we're going to have non-cellular, which will be our viruses. And then we have our prokaryotes, which are our bacterial cells. And we have our eukaryotes, which are going to be our cells that are more advanced, that have a nucleus and organelles. And we talked about all these in BIO220, but just briefly to summarize the difference. Remember, prokaryotes, they have one single circular, which I know it's coiled up right here, but if you could unravel it, it would be a circular chromosome. They have no organelles, they have ribosomes and a cytoplasm, but they don't have the complexity and the organelles of a eukaryote. Eukaryotic cells is what human cells are, animal cells, and they have a nucleus, they have all the organelles, they're much larger than prokaryotic cells. Talk about our classification of organisms. Remember the taxons: domain, kingdom, phylum, class, order, family, genus, species. You do have to have that order memorized, and you need to know what it means. This is how you classify all living things, and our microbes are no different. And just remember that, if you remember this sentence, dominating King Philip came over for good soup. And that tells you the order from domain down to species. Now just remember, domain is the least specific taxon. Species is the most specific. Think species specific. That's why it has that name. So here's just an example of what that would look like for a red fox. Domain Eukarya, kingdom Animalia, and as you go all the way down, you get to the genus species. And that is what is shared with an organism. You don't have to give the whole list. You would just write the genus species, capitalized genus, lowercase species, all of it written in italics. So Carl Linnaeus developed that taxonomical classification system. And in his, he only had 2 kingdoms. Today, we actually have 6 kingdoms. You do need to know the 6 kingdoms. That would be Plantae, Animalia, fungi, eubacteria, protista, and archaebacteria. And I know we covered all this in 103, so this is just recap, but you do need to be familiar with it. So kind of the progression of that classification. 1735, you have Linnaeus, the 2 kingdoms. 1866, protista is added. 1938, they added Monera, which was bacteria. 1969, they add fungi. And then 1977, Woese proposed that all organisms should be organized into 3 large groups called domains. So then we get the domain, you get archaea, bacteria, and eukarya, and the 6 kingdoms. So it goes domain, kingdom. And then in 2015, they added a 7th kingdom, chromista, or at least it was suggested. And there is Carl Woese. So 3 domains, archaea, bacteria, eukarya, and then you have your 6 kingdoms. So just make sure you have those memorized. You probably still do from BIO220. And then they have these super groups that they classify things into, and we'll actually talk about that some more later on. I know in 103 we talked a little bit about it. We'll also talk about that some. Just make sure you know also how to write the genus species for an organism. Remember, the genus is capitalized, the species is lowercase, and it's all in italics. All right, last topic here will be immunity and transmission. So immunity is the ability to recognize and repel disease-causing particles. So our bodies need to be able to tell what is ours and what is not ours. So what is self and non-self. So our cells have these groups of molecules or proteins called MHCs are major histocompatibility complexes, and those are used to identify each cell. So before a person is born, the lymphocytes or white blood cells roam the body, and they learn to identify the body's own cells as self. When a person is born and exposed to other particles, the lymphocytes are able to recognize them as not being ours. So non-self particles that are pathogenic, that's what's going to trigger the body's immune responses. So the MHCs, that's very important in preventing the immune system from attacking our own body. If something goes wrong there, well, then you can have some kind of autoimmunity where your body's targeting itself when it shouldn't be. We will talk about these in the immunology notes, but just to give you a brief sort of introduction, we have what's called innate defenses and we have adaptive defenses. Innate is non-specific. You are born with this type of defense. An example of this would be, well, you have skin. Your skin keeps things from just crawling right into your blood and infecting you. So there'll be a surface barrier, your mucous membranes as well, and like your nasal cavity and your sinus cavities. So that's your first line of defense, what things run into first. Other innate defenses that you're still born with, but they're internal, might be things like natural killer cells, having a fever, the inflammatory process, having phagocytes that can take a pathogen and just eat it. That's what those pathogens are going to come into secondly, but it's still an innate defense. It's not specific to certain diseases. It's just a general defense against whatever comes in to your body. Well, your third line of defense is going to be your adaptive defenses. These are specific. These are acquired over time. We can have humoral immunity, which is going to involve B cells producing antibodies that will help our immune system target foreign substances, foreign invaders. And then you have cellular immunity, which is going to use T cells that are going to attack those foreign pathogens. You do need to know, and we have covered this before in other classes, is the antigen-antibody correlation. But an antigen, that is something that triggers the production of antibodies. So you could think of antigen as a foreign particle. Antibodies is something we make. So antigens trigger the production of antibodies. And you'll notice in this image how the shape of the antibody is like an impressed triangle, and then the shape of the antigen is a triangle. So it has to be able to complement the physical shape of the antigen. So we can produce millions of different antibodies, and the specific shape of the antigen binding site has to correlate to the antigen, or it cannot interact with it. And it has to stick to it so that we can target it for destruction. And that's what a vaccine does, is you take a weakened, attenuated form of a bacteria or a virus or a microorganism, and you introduce that into someone's body. Well, then their body's able to create immunity for the particles of the microorganism without having the microorganism able to reproduce and cause infection in our bodies. So it just allows us to train our immune system. It's super awesome that these are developed. So a vaccine, you just take any microorganism or disease-causing agent, and you would weaken it. You could boil it, break it into pieces, and then introduce that into someone, and it won't cause infection. Some of the symptoms of that virus might happen because those particles are in their body, but it won't be as severe, and then your immune system can be trained around that virus, which is pretty cool. So different viruses have, different vaccines that we can develop from them. So you could have a virus that's based on the protein that a virus might produce in our bodies, and it's just a vaccine against that protein. So when you're exposed to that virus and that protein is made, you're able to activate and destroy it, or activate our immune system to destroy it with antibodies you've already made for it. You could have whole virus vaccines that are inactivated or live attenuated. You can have RNA-based, DNA-based, all kinds of different vaccines. So for our second line of defenses, you'd have inflammation and phagocytosis. So for that, if you step on a nail, it triggers an inflammatory response. And what will happen is neutrophils will enter the blood from bone marrow. We'll have margination. That's where those white blood cells are going to move against the walls of the blood vessels so that they can get ready for diapedesis, which is where they slide through and move towards the area of infection. They're attracted to the chemicals. We call that positive chemotaxis. So the microbes will adhere to the phagocyte, and it will ingest them. The lysosomes will release hydrolyzing enzymes that destroy the antigen. So our third line of defenses will be humoral immunity. So this is where B cells make antibodies. We'll talk about these a lot more later, but just to show you, you're introduced to a certain type of antigen. Your body produces antibodies that can then stick to those antigens and target them for destruction. Cell-mediated immunity. This is going to use T cells to directly kill infected cells and fight intracellular threats, viruses, cancer, and it involves a delayed response, but targets compromised body cells. So it's using targeting cells, and then for our humoral immunity, it's going to be in the fluids where this takes place, and you're producing antibodies. And there's some images of what that can look like. And we will talk about that more at a later time, so I'm not going to go super in depth on that right now. Then we have what's called BSL facilities. And what that stands for is biosafety level. So basically these are labs designed with specific practices and equipment to safely handle what could be extremely dangerous agents. So you have a range from BSL 1 to BSL 4. This is where all the BSL 4 labs are. And this would be the most dangerous, have the extremely high risk. And you'll notice there's some in Russia and Europe, excuse me. And then there's a lot in the United States. So it's ranked based on the severity of what you're working with. So for example, in Atlanta, Georgia, that's the Centers for Disease Control and Prevention, BSL 4 lab, and they actually have smallpox there. So that's where it is. So if smallpox breaks out, we're all not that far away from it. So anyway, think of it from no or low risk to high risk as you move from BSL 1 to BSL 4. Some organisms you might see in BSL 1 is yeast, right? Not dangerous. BSL 2, you might have measles, getting somewhat dangerous. BSL 3, you might have tuberculosis. And then BSL 4 is going to be the most serious. You might have like Ebola. For BSL 1, you'll have open laboratory benches. You'll wear PPE, but it's really like not any risk here anyway. So the microbes are not going to cause serious human health concerns in a BSL 1 lab. For BSL 2, it gets a little more serious. Use PPE. Might have coats. You might have to separate everything into safety cabinets. And then you want to have self-closing doors, as well as an autoclave to disinfect materials. BSL 3, PPE, safety cabinets. You'll have respirators, 2 self-closing locked doors, and you'll need filtered air in a BSL 3 lab. And if you're working with, tuberculosis, you're going to want to have that extra protection. And then BSL 4 is the most serious: air supplied, positive pressure suits, hazmat suits. You're going to have showers, decontamination areas, UV light room, and a vacuum room. And I like this image that was on this set of notes. It says no photos for the BSL 4 lab, but they took a photo of them. So that brings us to some general terminology that we'll look at. So symptoms versus signs. Symptoms are subjective. This would be how the patient feels, pain level, description of aches, things like that. So symptoms are subjective. Signs are objective, and they are measurable. So, for example, a certain sign of a particular illness might be a high fever. You can measure that and get a temperature. Or it might be high blood pressure, which could also be measured. So signs are measured. Some other terms. You'll have carriers. That would be a person that has the microbe, but no signs or symptoms of the disease. But because they still have it, they can transfer it or transmit it to somebody else. A vector would be an animal that carries the microbe and transmits it to someone else. We have zoonoses. These are animal diseases that humans can get. We have fomites, which are inanimate objects like straws or utensils that transmit the disease to someone else. Last terms, and then we'll be done. One of these is communicable. Communicable means infectious or transmissible diseases that spread from host to host through direct or indirect contact. Examples of this can include STDs, mononucleosis, chickenpox, influenza. Contagious would be a communicable disease that is easily spread from host to host, which usually means without direct contact. So it could be airborne. Chickenpox could be an example of that, or influenza. And then non-communicable would be diseases that are not spread from host to host. Examples would include heart disease, high blood pressure, for example. And that will be our first set of notes.